Hello, my lovely anatomists and physiologists. Let us talk about the very first step to our adaptive immune response, which is antigen presentation. And it makes sense in this conversation to begin by, okay, what is an antigen? What is an antigen? So let's talk about what we mean by an antigen first. An antigen is going to be a glycoprotein marker on the cell membrane that's going to be like specific to each type of cell. So glycoprotein means that it's a carbohydrate added onto a protein. So we can also think about it as a protein carbohydrate marker, right? We talked about this way back in chapter 3. When we talked about cell membranes and anatomy and physiology part 1. And when we talk about the antigen, we can also think about it as a marker that generates an immune response. And my understanding then is that this is part of how we're getting the name. In particular, the immune response that we're talking about is antibody production. So we're generating antibodies. This is the marker that generates antibodies. That's how we're getting the name antigen. All cells have them. So when we talk about self-recognition, our cells have glycoprotein markers and our immune cells are trained to recognize those as self, to not attack. So our glycoprotein markers are recognized as self and as such are not generating an immune response. So we wouldn't really think about them as antigens in that case. When we talk about antigen presentation, what we see happening, number one, is that I'm going to go ahead and say foreign antigen becomes internalized by the cell. And there's two ways that this happens. One way is infection. If a cell is infected by bacterium or by virus, then it has access to the glycoprotein markers on that particular cell. If the cell is a phagocyte, its whole job is to consume that foreign cell and do antigen presentation. So the next thing that we see happening, and I do have a diagram here that we're going to look at together. It looks kind of busy, so just sort of keep your eyes off that diagram for a minute if you can. The next thing that we see happening is the cell. So this could be your phagocytic cell, or this could be your infected cell, is going to break down the pathogen. And this is involving the organelle called the lysosome. And it's going to display the antigen outside of the cell using what are called major histocompatibility markers. No, complex. Major histocompatibility complex, which we often abbreviate MHC. Consider this like a flag off the cell, saying there's an antigen here. Hey, I found this. Hey, this is inside of my cell. This is inside of me. So antigen presentation is essentially a flag that's being held out by the cell that's either infected or the phagocyte to let our white blood cells know that we found it, that it's present in the body. Now, when we talk about the major histocompatibility complex, we see that there's two classes. So we have what we can call MHC1. So this is class 1. And then we have what's called MHC2. So this is class 2. This is super important. So let's highlight MHC1 and MHC2. MHC1 is like the first class. It's the standard. It's the basic population of your cells, is your body cells. And so what this is meaning, if you have antigen presentation using an MHC1, using a class 1, this means you have a cell that's infected. You have a cell that's infected. And if your cell is infected, it's a dead cell. It's a dead cell. We can't take a tiny little vacuum and suction out that virus or suction out that bacterium. Instead, we just kill the cell. And if we kill the cell, we're killing that virus, we're killing the bacteria also. So when we talk about antigen presentation using class 1 MHC proteins, this means our cell is infected, and ultimately, this is going to stimulate what's called a cytotoxic T cell. Cytotoxic means toxic to cells. This is a hand-to-hand, cell-to-cell combat cell, and it is deadly. So what we see happening is if you're presenting an antigen with one of from your body cells using your MHC1 markers, that means the cell is infected and the cell will be killed. When we look at the MHC2, on the other hand, this is a special class of cell. We can think about these as professional antigen presenting cells. And we're going to abbreviate antigen presenting cells as APC. So we're going to find a class 2 marker on this special population of cells, which are professional antigen presenting cells. And these include macrophage. These include dendritic cells. I mean, y'all look, these are your first line of cellular defense. These are your phagocytes. And these include your B cells. Now, I really like this idea of a professional antigen presenting cell, meaning it's the job of this cell to identify foreign invaders, gobble them up, phagocytosis, and then display the antigen, triggering the adaptive immune response. Having an antigen presentation with class 2 MHC is a good thing. That cell is working. That cell is effective. You do not want to kill that cell. You do not want. Another way I like to think about my class 2 MHC protein is like a gorgeous dog, and you're playing ball with the dog, and you throw the ball and the dog runs out and grabs the ball and then brings it back. That's what that dog is supposed to be doing. So you pat the dog, you say, good doggy, like, well done. Good job finding the pathogen. Good job showing me the pathogen. Now we can respond, right? Or like the dog is barking that you have an invader at your house, right? Like the dog is doing its job. Like that's what you want the dog to do. So ultimately, what we see happening as a result of antigen presentation on class 2 MHC proteins is antibody secretion. So the cell that's presenting the antigen is not killed, but it just triggered our antibody secretion. So this is super awesome. Okay, let's take a look at our diagram. And we can see that our starting point is down here. And there's lots of words on the screen, but what I want you to pay attention to is, okay, so let's take a look at this cell. This cell has a big body, and then it has like a ton of these extensions, these cellular extensions. Looking like cilia extending from around the whole cell. And what we're seeing is that, you know, this cell is doing phagocytosis. So it's actually engulfing a pathogen and bringing it into the body of the cell. And what we see then is the lysosome, that organelle fuses with the vesicle that has the pathogen inside. We see that we're breaking down that pathogen into little pieces. Those are the antigens. We're combining those antigens with our MHC proteins. And because the cell is doing phagocytosis, then we are showing out using class 2 MHC proteins. And then we're seeing the actual presentation here of those MHC proteins displaying this antigen that we found. We're seeing that happen in 2 ways. So we're seeing another example over here of antigen being engulfed or phagocytized. We're seeing another area where we're making those MHC2 proteins. And again, we're seeing them being displayed as flags. So this is what we mean by antigen presentation. Okay, stay tuned. We're going to talk about our T cells just a little bit. And as always, take care of yourselves and each other.